these are H3 influenza viruses from, well, this particular set. All of the small ones in the top left here are from July 2022, so isolated from somebody's throat in that month, tested antigenically, in this case, against something like 15 or so different ferret, primary infection influenza, antisera. The big blue dots are vaccine strains. The ones that are buried here in this antigenic cluster are ones that were used at that time. And these ones here, these ones are going to evolve in the future. And I just want to show a little representation of the evolution of these seasonal human influenza viruses. It's a similar gig with SARS-2. 1 year later, you see, we're in July 2003 now, and the viruses have moved on to this next antigenic phenotype, and that's enough of a antigenic change to necessitate an update of the vaccine strain. The grid in the background is a twofold dilution, in this case, in the hemagglutination inhibition assay. You see there are still a few trailing viruses still in the previous cluster. Here, but and a few stray outliers. 1 year later, 18 months later, the virus had moved on to another antigenic cluster, necessitating another update of the flu vaccine. It's a very busy time for the evolution of the virus. Indeed, 1 year later, within all of this, multiple lineages like Richard described yesterday, and like we see in SARS-2, co-circulating lineages, multiple different amino acid substitutions, and then the virus stayed in this cluster for about 3 years before moving on to the next antigenic cluster. This is a little bit further back in time from that. These are the H3 influenza viruses from 1968 at the top through to about 2002. Oh, I forgot to also say in these antigenic maps, they're constructed from these titers that are translated into distances and then reconstructed like one used to in cartography in the old days before satellite imagery. And the X and the Y axis here, well, it's just like on a regular map of DC or wherever. They're both axes of what a mathematician would call distance. In other words, this is rotation-free and reflection-free. We could tip this map upside down. The relative positions between the variants would be the same. Tip it by 9 degrees, whatever. That's how to interpret this. And when points are close to each other, that indicates antigenic similarity, as I imagine you already know or would have guessed. So similarly here, a whole series of antigenic clusters, the average time between the virus jumping from one cluster to the next, necessitating an update of the vaccine strain, being about 3 years during this period. It's still pretty much approximately the same today, although as we saw, sometimes it's faster, sometimes it's slower, driven by super interesting reasons that are really nicely being worked out these days, although the field is not exactly completely on top of it. You notice here these names. When thinking about the antigenic phenotype, the thing that has been useful for many years is to talk about strains that are like a particular vaccine strain. So at the top there, that's Hong Kong, 1968. The next one, England, 1972. The next one, Victoria, 75, Texas, 77. These were the vaccine strains that we use in those clusters. And this abbreviation is something that we used in this paper, and people have subsequently used when talking about these clusters, when trying to understand the fine details of this evolution. And of course, at this level of granularity, this is about enough antigenically, but some of these clusters have little mini cluster transitions within them. I'm not going to take you through all those details now, but that sort of stuff happens as well. And like many people mentioned yesterday, it depends what level of granularity one wants to be at. Well, and I already talked, yes. So if one is vaccinated, for example, with a virus in the middle of one of these clusters, one will typically have immunity against the rest of the strain in that cluster. Maybe in some instances, if it's a particularly potent vaccine, or if it's a particularly good infection somebody's had, that immunity will be a little bit broader. And so you can see here that when the virus jumps to a new cluster, that's when it's necessary to update the vaccine. I'm next going to talk about how sometimes it's just a small number of the amino acid substitutions that get fixed over time that matter. This table here is the number of amino acid substitutions between each of these antigenic clusters. So, for example, between the top two clusters, there are 5 amino acid substitutions that are between every pair of amino acid every pair of viruses in that cluster. You pick any 2 viruses, there's typically more, but it's at least these 5. And so on for the next cluster. You see sometimes it's 13 or 14, sometimes it's a smaller number. Bjorn Kohl in Ron Foucher's laboratory set out to look at the genetic basis of this antigenic change, determining antigenic and genetic consensus viruses for each of these clusters indicated here by these green dots, and then measuring, looking to see which of these individual amino acid substitutions actually were responsible for the change. For a long time, people would say it takes at least 4. Well, it turns out that it usually takes just 1, occasionally a couple, and you can see here which ones they are. Oftentimes, it's amino acid substitutions at the same site or at a relatively small number of sites. In fact, when Bjorn mapped these particular locations onto the HA structure, here the HA structure, the transmembrane domain at the bottom here in trimer form shown by the light, medium, and dark grays. The receptor binding site is up here. And marked here are the 130 or so surface amino acid locations that are in the 5 antigenic sites. And what Bjorn found is that the ones that are critical for major antigenic change as seen by ferret sera are these green ones around on the periphery of the receptor binding site here shown in yellow, and a little bit of a couple of others here that are also important, but in a less critical way than the ones for this data set that were in green. Nicola Lewis with collaborators subsequently showed that for equine influenza as well, and along with Amy Vincent and other colleagues in the USDA, showed that for swine influenza virus as well in most cases. Interestingly, a little more complex for avian influenza viruses, a little bit further away from receptor binding. So I'm showing you this to say, yeah, all of this genetic evolution, fixation, different lineages can happen, but major doesn't even just boil down in the case of flu for this antigenic change to one of the surface protein. It actually boils down usually to a very small region of that protein right around receptor binding site. It's a similar thing for SARS-2 as a SARS-2 antigenic map, data from David Montefiore's lab at Duke, pulled together by Sam Wilkes and Barbara Meulerman. On the left-hand side here, 614G, alpha, major gamma, and don't forget mu Betty up here, major delta down here, major the ones that many will have embedded into their retinas, into their minds, and the early Omicrons here. And similarly, when looking at which amino acid substitutions are responsible for antigenic change, it is usually, so this group of viruses that are over here to the right of the original virus, it's almost certainly just the substitution at 484 that causes that antigenic change. These viruses down here, it seems to be a 452 substitution. And then, of course, to Omicron as well, but a low Omicron had many amino acid substitutions from the original virus. Just a couple of those seem to be the ones that cause the major antigenic change, as shown very nicely by, for example, David Ho's laboratory in Columbia. Major. To take this data from David's laboratory on a little bit further in time, these are data from Yunlong Sao's laboratory in Peking University in Beijing in China, with a mouse sera antigenic map. The one from David Montefiore's lab was human sera. Major 614G here. You can see how it corresponds to the antigenic map with the human sera in David's laboratory, followed by the early Omicrons here with and then what we call anyway the late Omicrons, and now of course BA286 followed by JM1, and as Betty so beautifully showed yesterday, the major institutions after JN1 as well. Of course, when talking about this sort of in retrospect, with thinking about this broadly and where the vaccine strains were, this is a pretty good level of detail. We can pretty much get this. It's a good aid memoir, but actually at the time, there's all the sorts of detail that Becky major yesterday, and also a lot of antigenic detail as well going into the vaccine strain selection process. Okay, and I should also mention here's the SARS-1 virus, which interestingly, although quite diverged antigenically, is also antigenically not completely distant here. It's a similar story, for example, for the MERS coronaviruses. Ralph Barrick is doing a very nice analysis of different MERS coronaviruses to see just what sort of cross-reactivity there is. Of course, this is useful not only for doing vaccine strain selection at the time, but also for thinking about doing preliminary work to generate vaccine candidates to prepare for maybe a MERS coronavirus pandemic, etc. And also for thinking about major virus vaccines. Well, yeah, what should they be tested against? Well, you could test it against a whole bunch of viruses, and ideally what you want is representatives of different parts of antigenic space. And so that's why trying to link up across, for example, the MERS coronaviruses is important for knowing which ones to put for testing viruses to prepare for outbreaks in the future. The last thing that I want to mention is that here what we saw was that it's typically just one or two amino acid substitutions that cause these antigenic cluster transitions for that necessitate vaccine updates. Sometimes it's a much deeper genetic divergence with many, many more amino acid substitutions, and sometimes not that much antigenic change. Here are some representatives chosen representatives over time from many dengue viruses by Leah Katzelnick, leading this many titrations in Steve Whitehead's laboratory here at NIH. 4 different dengue types. And if one does the same sort of map making on the genetic data as one does for the antigenic data, you see that these 4 different dengue types are highly distinct and highly related to each other compared to the depth of the genetic distance between them. And of course, one sees that in the tree in the left. Here on the left is that same genetic map of the influenza viruses, and on the right, a antigenic map of those viruses. And you see that things are far less distinct. And similarly, that although it was previously sort of the received wisdom was that there are quite distinct dengue serotypes, we see, for example, here there are some different dengue types that are more similar to each other than there is diversity. Well, I should say the diversity within some of these types, for example, DENV2 here, is just as great as the distance from DENV1 to DENV2, and similarly with DENV3 and DENV4. So if one, for example, is doing vaccine strain selection here, we really can't be thinking, okay, just one representative from each of these dengue serotypes. We need to be carefully choosing which one. So 4 strains in the dengue vaccine, and depending on particular ones that one would choose, one would get more or less coverage. So this again speaks to going sometimes into greater detail. And the final thing that two things to say. Back to flu now. This is work by Adinda Koch, Ron Fouché, and Mathilde Richard at Erasmus Medical Center in Rotterdam. These are the H5. This is the H5 evolution from, pretty much through all of the H5 viruses. Justin talked about this very nicely yesterday. The same sort of clade names as he showed from that nomenclature paper that came out in, when was it, 2007 or something like that. And the different colors here on the tree showing representative viruses throughout the tree that Adinda, Mathilde, and Ron reconstructed to make an antigenic map of all of these viruses measured together. That map is on the right. The coverage. Same coloring is in the tree, and you see this whole thing is overlapping. Occasionally, there are distinct viruses, like these clade 1 viruses over here, the yellows. Coverage. Most of the 2.2s in deeper red here. There is one over here, and then some coverage. The 233s and the 2344s. Sorry, the 2344s and the 2344Bs. These viruses down here covering much larger parts of antigenic space and spanning large regions. And indeed, some of these viruses, although antigenically substantially different from the earlier classical viruses, actually ending up antigenically very similar. If we think about vaccine strain selection here, there have been something like 40 now different vaccine candidates as the viruses incrementally evolved. And if one is just tracking the genetic evolution and thinking about this, indeed, there are many different variants that have come up. Coverage. One's just looking, for example, within a particular genetic clade, and then, look, here are some interesting changes. Test them in HI, look locally, and there are indeed changes. It's important to have a new seed virus to be ready to go. But actually, when one looks at it like this all together, coverage, the size of this antigenic space could probably be covered by 4 or 5 different variants if they're very carefully chosen. Again, an important thing when thinking about what viruses to stockpile for coverage, for preparing for a potential coverage. And the final thing that I'm going to say is to come and to talk about the swine viruses. Tavis talked very well about the swine viruses yesterday. I'm going to talk about just one of them. It's the Eurasian swine H3 viruses. We see here this same antigenic map that we saw of the seasonal human influenza viruses, from the Hong Kong 68 viruses down here through the 2002 viruses, multiple updates of the human virus vaccine. And in green here, the Eurasian swine viruses that have been in swine from humans since about the mid-70s by a virus similar to one of these viruses indicated by the arrow here, got into swine. And this is the evolution since then. And you see hardly any antigenic evolution in these pigs. If one looks genetically, coverage. So here at the top are different influenza viruses from the first couple of antigenic clusters of human seasonal viruses. Here are other seasonal viruses from flu going through to 2002. And here are a bunch of swine viruses during that period as well. And you can see that from a phylogenetic standpoint, there's hardly any difference here. Coverage. The key differences are that these substitutions, the clade structure, all of this genetic evolution is not happening close to the receptor binding site. And so there's hardly any antigenic evolution in the swine viruses, almost certainly because there's no need for it. And the viruses are, pigs live for about most of them live for about 6 months before some of us eat them. And so there's always naive pigs, less pressure on the virus to evolve antigenically. And so I wanted to finish with this antigenic map where we've essentially got the same virus doing pretty similar thing coverage genetically and phylogenetically, but doing very different things antigenically. Coverage. And of course, the stuff that Tavis talked about yesterday is not just this. And I know that, but as he talked about yesterday as well, there is antigenic diversity in these swine viruses. In Northern Europe and in many parts of Asia where these viruses are circulating, there is no vaccination. But like in the US, you vaccinate, coverages diverge. And we need the sort of stuff that Tavis talked about yesterday. Coverage. So these are the 3 points that I wanted to make. And then the final one is just to say that one of the things we did in flu for a while, and it was pretty useful, was to use a dot in the genetic nomenclature, such a year like 3C3.2a, to indicate the moments when there was an antigenic change. Coverage. That's it, other than, as you can imagine, many, many acknowledgements. And particularly Ron Foucher's laboratory or sister lab the last 20 years. Thanks.
Thank you. Our next speaker is Peter Simmonds from University of Oxford, and he's going to be talking about virus classification and support for different stakeholders.
Great, thank you. Fantastic workshop, amazing to meet a lot of people I know by name but never actually met in person, so fantastic opportunity to do a bit of networking. The meeting as a whole is really quite diverse in what it's trying to cover. When I was asked by Elliot to talk about stakeholders and so on, I went into the idea that we're going to talk about taxonomy, formal taxonomy, and the way in which it can be potentially applied below the level of species. However, when we listened to talks yesterday, we heard about evolution on timescales of 1 or 2 years. Okay, so there is a sort of, we have to think about a bridging point between sort of top-down taxonomy, where we start off with species and go downwards, and actually the very dynamic and very lively evolution that actually occurs in the very short term. And as Derek and others have shown, often in response to immune pressure and immune escape and so on, which are not long-term evolutionary drivers. So anyway, with that sort of preamble, really just to try and bring over the concept, I'm not going to be talking about the same thing. I'll just go through a few slides talking about stakeholders and where this impinges on sort of classification decisions and what ultimately the classification is actually for. So I've got rather wordy slides. I'm not going to go through all the words here, but I mean, I just wanted to firstly list who the stakeholders are. And this is a list I kind of came up with. I may not be completely all there, but obviously the main stakeholder in classification is ICD members. But putting that to one side, obviously the people who use classification are clearly the wider virology community. There's obviously a science base around virology where we have evolutionary biology. We want to understand origins of viruses, their genetic diversification, and so on. We also have, however, outside of this sort of scientific area, educators and people teach virology. We need a stable system to explain how viruses are grouped into families and so on. And we don't want to change that too much over time, as if we can possibly avoid it. Major importance are obviously the clinicians, public health, who actually use virus categories in their descriptions of disease, but also in terms of virus spread and giving labels to emerging viruses and emerging strains, as you've heard in the last day or so. And then finally, we have regulators, and clearly we have biocontainment restrictions on viruses. And if you're going to say that this virus is a Cat3 or BSL3 pathogen, then obviously we need to have a proper description of what the virus is. So poliovirus has a completely different set of restrictions from what actually are quite closely related enteroviruses. Similarly for HIV and immunodeficiency viruses from chimpanzees, which actually are also, again, very closely related. And then I guess, and we'll come on to this, SARS-CoV-2 is a completely different category from sarbecoviruses infecting bats. So obviously we need some sort of system where we can actually define groups for biocontainment and also for export-import regulations. And of course, the plant virologists are very concerned about defining virus species or below species levels so that they can safely export potatoes from Brazil to Europe, where, for example, there may or may not be a particular pathogen present. So that they are very aware of that. And clearly this is an important aspect of how you would classify viruses. So that's sort of the background. I'm going to try and now show how these sort of stakeholders impinge on various aspects of classification of viruses. So first we're going to talk about species. Okay, so I think if we're going to talk about below species classification, we better understand what species are. And unfortunately, this isn't an area. This is an area where virology has somewhat diverged from classical biology. I mean, necessarily, obviously, because as a very broad generalization, the original definition of species in biology was defined by interbreeding capability. I know that's very old-fashioned, but essentially that is the conception behind a species. It's the lowest, the divisible unit of an animal or a plant where you can show interbreeding capability. And of course, that then defines common gene pools and gene exchange by meiosis and so on. So that's all quite logical. The problem obviously is that bacteria and viruses don't have sex in the same way. So we need obviously a different definition for that, even though we might use the species term for those. Bacteriology have their own issues, and I won't go through those, but for virology, we essentially have a sort of somewhat descriptive and more recently genetic basis for assigning species. And this is the current definition, halfway down the slide. It's the lowest taxonomic level in the hierarchy approved by STB. Not that's a particularly good definition, but it's a monophyletic group, so we can't have polyphyletic categories, which is fair enough. And then it's vaguely specifies multiple criteria. And of these, it is genetic divergence that is increasingly used to define species, particularly now as we can assign species based on sequences alone. We don't need to have a list of cell culture properties or lists of diseases associated with infection. So that's been a sort of transition. We had a few hundred species when I joined ICTV. We now have over 13,000 species, almost all of which are now genomically assigned based on metrics of sequence similarity or clustering on phylogenetic analysis. I would just make the point now that we have an absolute definition of species in general biology in terms of interbreeding. The level at which we assign species in viruses, though, is somewhat arbitrary. Okay, and it's based on clustering. And there is huge variation in the sort of divergence thresholds where you might decide something is the same or different species, as I'll show you in a graph later on. Okay, so in terms of its descriptive ability for stakeholders, we have several situations. And if we go back to the origins of virus classification, we've typically had something like this, which is a sort of one virus, one species concept. So, for example, there was an infectious agent that caused yellow fever in parts of Africa. The phenotype, disease phenotype, was transmissible. We could actually see the same disease where the virus spread. And so it was quite reasonable to call the virus yellow fever virus. And you can make that a species. Okay, and actually, a lot of the arbovirus classification is based around disease descriptions like this. And essentially you've got one virus in a species, which is fair enough. And actually that's exactly what most of the plant viruses are classified as. So we have amazing descriptions like banana bunchy top disease, which actually is caused by banana bunchy top virus. And we have carnation mottle virus and so on. So these are very descriptive. Of course, when you do the genetics, they are obviously different viruses, but the assignment of a species to that virus is pretty much fixed. So that's kind of where we were in the, I guess, 70s and 80s. And of course, the genomics came in at that time. We now have situations where we have species which are kind of an overarching category where we may have quite different viruses inside the species. And they've been kind of formed genetically. So, for example, the enterovirus Coxsackie pole species in blue, which was it used to be enterovirus C until last year. That contains all sorts of viruses because genomically they're all closely related to each other, and you don't want to put them apart. The difficulty, though, obviously, is that the properties of the viruses within the species varies considerably, and I'll be using the example of poliovirus in other parts of the talk. Clearly, poliovirus behaves differently from some of the Coxsackie viruses, which actually are also classified into the species. Okay, so I put a yellow exclamation mark as to whether this is satisfactory or not. But what is very unsatisfactory is something like this, okay, with the use of the term HIV-1, okay, and actually HIV-2 as well, because clearly we are making a humans, but actually the viruses that make up HIV-1 are polyphyletic. And in fact, there have been 3 zoonotic transfers of chimpanzee or gorilla viruses into humans, which actually set up the main group, O group, and N groups, and actually P groups as well from gorillas. So as an entity, they are, you know, the cause of AIDS. They have BSL-3 restrictions, and we have AIDS conferences and so on. But of course, this is polyphyletic because within those we have SIVs, which we would probably handle at level 2 in most labs. And have quite different tropisms and properties and so on. For example, SIV in chimpanzees doesn't cause AIDS. So that's an example of where we must have a clinical category, but actually it's not supported by a species definition. And then finally, we can get into things which are recently zoonotic, and SARS-CoV-2 is a good example of that. We have to have the category SARS-CoV-2 because it is obviously essential for public health, for all research done on the COVID outbreak and so on. But of course, it's a very, very much clustered into bat subecoviruses. And in fact, this tree here from Dave Robertson's group in Glasgow kind of just depicts the species as a whole. And of course, they show bats as hosts, but of course, at one point there is the zoonotic SARS-CoV-2 and the SARS-CoV down here, again, very closely related to bat viruses. So again, it's a category which isn't supported by a species assignment. Right, so the other point obviously is we can classify viruses in quite different ways anyway. So we are from textbooks, and when we first did biology, probably familiar with the Baltimore classification. So this classifies viruses based on their expression strategy of genes. So if that's the RNA template for protein production, then this is derived in multiple ways from different genomic configurations. So a plus strand DNA might actually replicate and actually act as a template for a plus strand messenger RNA and so on. And then of course, there are various ways in which DNA and double strand RNA genomes can actually generate that template for translation. Problem is though, that in terms of its genetics, then these are not categories that are supported by evolutionary descent. And at the moment we have a kind of classification of viruses into 6 realms, which are ultimate origins of viruses. And actually it turns out that the Baltimore Group 1 comprises at least 4 of the 6 realms, whereas the Baltimore classes 3, 4, 5, and 6 are actually all 1 realm, Ribaviria. Okay, so that doesn't match, but nevertheless, the Baltimore classification is still important. It's not undermined by an evolutionary treatment, but nevertheless, it does represent an alternative classification that is useful for certain purposes. So I think we have to bear that in mind. And of course, we should, we have terms like arboviruses. That's very useful for describing the characteristics of actually many different groups of viruses in the evolutionary terms, but which actually have a shared vector-borne transmission route. And we have neurological viruses, cardioviruses, and so on. So again, we have clinical categories which are polyphyletic, but nevertheless can be useful as descriptive terms. Going below species is something where I mentioned already that, at least in the ICV code, the ICV does not classify below the level of species. So I think a lot of the stuff we've been talking about in the last day or two is not going to appear in the ICTV master species list. And it's not going to be treated as taxa in the way that species and above would be. Nevertheless, below species taxonomy is quite common in the rest of biology. And of course, we have established subspecies systems for zoology. Pan-troglodytes, troglodytes, it would be one, contrasting with Virus in West Africa. And of course, these are not biological species in the sense that those two subspecies can interbreed, but nevertheless, they do refer to phenotypically distinct populations. And of course, in botany, they go even further. So they actually have not just below species categories, but different types of categories. So you might have varieties, subspecies, cultivars. And in some cases, you can actually have 2 ranks below species as well. So here's one, which is, I won't try and explain what that is, but a friendly botanist actually gave me that example of a form and then a sort of cultivar below that. So it can be complex. Virology just won't go in for that. And of course, they're therefore unrecorded formally in the ICV records. And they're the definitional way in which you would actually define categories below species of viruses is very much based on the wider community to develop a consensus on that. And of course, I've been involved for many years in the genotype classification of Hep C, which again is below species, it's not official. But obviously it's something that's maintained. We try to keep a consistent nomenclature for that. And again, it's very similar for enteroviruses with poliovirus and so on. I think in red, though, I could just point out that there are problems with that in the sense that we've already alluded to the fact that you might publish a sort of system of classification, but then it gets fixed, okay? And then obviously, if it develops further, then of course you'll need updates into the classification going into the future. So publishing papers isn't necessarily the ideal way of doing it. And of course, it may then depend on maintaining community websites to actually provide that information. The other point, obviously, is again mentioned yesterday, is that there are different classifications for different purposes. And of course, some get outdated and so on. So there are multiple classifications of many of the different virus groups. I think the other thing I just want to, again, just spend the last couple of slides talking about is the arbitrariness of the species level assignments. In contrast to biology, where we put that threshold for making a species the same or different varies considerably between virus groups. And to illustrate this, I'm going to show some results from PASC, which is actually, says when in Rome, behave like a Roman, but PASC was developed here. It's a very useful tool. You've actually seen it yesterday, but this is actually its application to the family Picorniviridae, where again, it records a sort of past distance, which is, I guess, something like percent similarity or ANI. And it has ranges for comparisons within species, between species or within genera, and then between genera. And these are quite distinct. So in fact, you can have an assignment threshold here where things that are more similar to each other than that should be in the same species. And if it's above that, it'll be a different species. So that's fine. However, I spent a bit of time just looking at PASC results for the main virus groups. And the problem there is obviously the level where it's set is somewhat variable between different virus families. Okay, so at one end, we have iridoviruses where genera are about 88% similar, and species about 93. And right down the other end with Hep C, species actually show divergence within species divergence, which is greater than divergence between genera in a different family. So that level is arbitrary, okay, and it actually goes all the way up from less than 70% right up to 98%. Okay, so we're not really looking at the same thing when we say an iridovirus species and a Hep C species. And of course, the way they've been assigned is just simply historical. It's somewhat based on groupings, but it's also based on classifications that were based on phenotypes originally. So that's kind of what we ended up with, and it's difficult. It contrasts a little bit with bacteriology. As I say, the bacteriology community have a somewhat phenotypically based classification of bacteria and type specimens, descriptives, definitions, and publication precedents. It's quite different from how virology works. But actually, if you look at their average nucleotide identities, then actually viruses that are called the same, have the same binomial name, tend to actually be more similar to the ones with different names. And actually, there have been various proposals to put thresholds for identity around the 94 to 96 mark, so you can actually make a genotypic classification of bacteria. And you might then expand from the 13,000 there are at the moment to something around about 1 million that are known from genomic sequences. Okay, so that may well presage a complete revolution in how bacterial taxonomy is done. Okay, so arbitrariness in action. But what I want to show you is something really horrible that revolves around thresholds. Okay, so we have, and this is an example of tree. There's a massive tree for coronaviruses. We have, I don't know, 60, 70 genera. Within the genera, we have dozens of species often in each of them. And then within each of the species, we have dozens and dozens of types. Enteroviruses, for example, have about 180 different types associated with the genus into various species. So it's sort of a hierarchy, but I can't show the whole tree, but essentially we've got a family level division into these different genera here. So we have enteroviruses, which I'm going to show you, but also obviously there's also other ones like hepatovirus, Hep A, paracaviruses, aphthoviruses like foot and mouth and so on. And then within enterovirus clade, we have species. These are the species A, B, C, D as of last year, but now renamed as binomials. And then, of course, within the species, we have a multitude of types, okay? And I've just colored these red or black because some are pathogenic and some aren't. And as I mentioned earlier, within species C, we have polioviruses, which obviously have entirely different restrictions, and they have vaccinations strategies, which obviously are completely irrelevant for these other viruses here. So these are essentially non-pathogenic, okay? So there is no point saying you're infected with species C, because that tells you nothing. You have to say that you're actually infected with genotype, with poliovirus or coccidic A22, if you're going to make some relevant clinical comments. And just in terms of where that threshold is, the coronaviruses are actually one of the more divergent ones. So these thresholds for species are set very high compared to a lot of other viruses. And in fact, frankly, there's no reason why you can't do that on the right in green, which is simply say, well, actually, I don't accept this threshold. I'm going to say, actually, the species level should be here. And these actually are genera, okay, and that's family in that case, okay, and that's perfectly within the range of other virus groups that there might be classified already. And all we do is simply rename them, okay, so we can have entroviridae, and then we can have alpha Kaksaki virus as a genus. And that means then that we can make all of these species if we want. Okay, so enterovirus 71 becomes a species, which is fine. Okay. Okay, so I mean, that's how arbitrary it really is. Okay, and I'm not proposing we do this, but nevertheless, I think it does show, however, that the categories we have are not robust in the way that we might have in the rest of biology. Where it manifests itself are problems like this. And in fact, Derek Smith will have, yes, okay, just one more slide. We'll have come across this particular document, which is our UK containment guide for viruses. Okay, and I've just reproduced the page which shows the containment levels for enteroviruses. Okay, and this is a document that's been written and updated for decades, and it has a very expert committee, but it is complete nonsense how it's actually been written for this. It's trying to use species, but then it realizes actually that a lot of these things aren't species. So, for example, there's all sorts of odd references to poliovirus here. Of course, for the bacterial pathogens, it's a clear, straightforward species list and subspecies as well. But for viruses, it gets itself into complete muddle. And I won't go through the errors here, but the black lightning bolts are where there are completely unacceptable errors in terms of the way the viruses are referred to. I can send this out if people want to have a look. But, you know, this is a real document. We have to deal with this when we actually make decisions about containment. And it is totally inadequate, not just for this, but for other viruses than this as well. So clearly the message about, you know, how we classify viruses has not got through to the regulators, at least in this case. Actually, in the States, there are similar problems, but of a different sort. Okay, so the other point is, again, if we want to maintain a classification, for example, this is a one page out of literally hundreds on the Cornivirally website, where a guy called Nick Knowles, who works in Purbright in England, has spent his life maintaining a classification of types and nomenclature for those types. And he maintains this website, which actually comprehensively lists all the different types. So this is species A, and these are all the different serotypes associated with it. He's about to retire, okay? We have no idea how this website's going to carry on. It's actually handwritten HTML code. So we don't know. We have no record. I mean, you know, he does it, but actually that's not going to work in the long term. So I hesitate to show the slide because actually this is a matter that's not been discussed with the ICTB. But I think one idea might be that actually we do take some steps to advance the formal classification of viruses below the species level because of this issue of arbitrariness and of also stakeholder usefulness as well. And I don't think we really want to start going down the route of renaming everything. And I think we should keep the species level as it is and don't make enteroviridi, for example. But nevertheless, I think we should formally recognize categories below species as taxa. Okay, and I wonder whether we could borrow something like the botanical nomenclature, where in fact we can have designators. So we could have type poliovirus 2 within that species here, and then we could have subspecies perhaps and isolates and so on. This would actually give a sort of formal definition for those, which we could define. There'd be in the ICTB records, and would then obviously be maintained and disseminated in a much more effective way than is at the moment with very much ad hoc community arrangements. And of course, ELIT could then add all these taxa to the master species list so they'll be accessible. And of course, ICTB actually brings advantages of permanence and accessibility and so on that would be missing elsewhere. And of course, we could supply the designations to GenBank and elsewhere so that they could actually add it to the taxon string that describes the viruses, so you could then search for them properly. Very difficult to search for categories below species on GenBank, as you may have noticed. So that's the sort of provisional proposal that we may discuss this summer in the ICTB meeting. And I very much hope that we can actually make some progress in that area. So this is just a summary, really. There are a vast number of stakeholders with differing needs, and we need proper descriptions, we need accountability, we need permanence and stability in how taxonomy works. And I think we should be very cognizant of the needs to go below species for many categories and for regulatory and medical purposes. So I'll stop there, thanks. And sorry, it's a bit long.
Coverage.
So, Speaker, I think this last slide might be a good template to keep up for the discussion going forward.
Coverage.
So I'll ask our two speakers to join me up here. And we might run a little bit longer into the coffee break. We'll see how the questions go.
Sure.
But there's some slack in the system, I believe. So is there any questions from the speakers from the floor there?
Right. I'm Elodie Gedden. I'm in the Laboratory of Parasitic Disease at NIH. So, Derek, I have a question for you. Something struck me while looking at the maps. So the maps are basically distance matrices. And so when you were showing the SARS-CoV-2 with the human sera and the mouse sera, it struck me how depending, I know this is nothing new to you, but I'm just wondering if we're going to use phenotype to help in classification. In this case, how the mouse sera were showing
Coverage.
BA2 and BA5 were in the same cluster, but in the human, they were very separated.
Yes. Coverage. Coverage. Sometimes there are differences, for example, as well with pseudotype versus live virus, whether or not a laboratory uses, overexpression of TMPRSS2 or ACE2. Coverage. Yeah, it's a dirtier process for sure, doing antigenic characterization. One also has to check with the maps that there are enough distinct and separate sera to get good triangulation of the points. Of course, when one does sequencing, modulo, the primer's working just fine. Coverage. It's a much cleaner world to be in. Coverage. Coverage. Yeah, it is a dirtier world. It's a world that has to be addressed, though. It's one of the it's a primary aspect of phenotype for some critical questions like the vaccine strain selection and some detailed investigations of how the viruses are evolving. But one has to deal with the mess. Usually it's okay. Coverage. Usually it's okay.
Can I ask actually how that scales across viruses? So you showed dengue on a grid, and then you showed flu before that, and then SARS-CoV-2 afterwards. Is the grid scale the same, or is it meaningful to talk about antigenic distances between viruses?
Well, Peter already knows the answer to this because okay. Well, we were on a UK committee together.
Oh yeah, right.
We at least brought up the question.
Yes.
Surprisingly, it does seem to. So those circles that I drew were about what current generation vaccines will give immunity to, and they are about the same.
Okay.
Yeah. And those different, and if one looks as well, not only with dilutions in the assay, that a twofold dilution in a VN assay for SARS-2 is about the same antigenic distance as for flu. When one connects that to the functional, can one be reinfected? Coverage. And that, the number of grid squares, the number of twofold dilutions for flu to the point when one can be reinfected, that's about the same, for example, as the original Wu1 SARS-2 to the early Omicrons. Yeah. And then similarly, the early Omicrons onto the late Omicrons with XBB, for example, as a representative of those. So surprisingly, yes, although no real reason why it should be. Coverage.
This question's for Peter. I was wondering, to those of us who are naive about the way the ICTV operates, if you could just describe that a little bit and how many people are involved in these decisions.
Actually, Jens will be talking about ICTV organization after the break. But I mean, very briefly, there's it is a sort of quite structured organization. So it has a president that has an executive committee, which actually meets once a year, which actually reviews proposals for taxonomy changes or additions. There are hundreds of those every year. And these are sent in by the study groups. There's about 80 or 90 study groups, mainly representing the main families of viruses. They themselves have memberships of 5, 10 or more virologists working in that area. They obviously overlap in membership a bit, but actually the size of the community involved in the study groups is huge. And in fact, around about a third of all virologists have been associated one way or the other in the study groups. So they generate the taxonomy proposals, go through to the executive committee. They're reviewed.
Coverage.
There's a decision to put forward those which are considered reasonable to a ratification vote, which is the entire ICU membership, which happens the following spring. And then once those are passed, then they go into the official taxonomy records. Actually, Elliot's just about to do this for this year's ratification vote.
Coverage.
Where we will have an awful lot more species and
Coverage.
Okay, so it's very structured. It's completely different from how the other codes work.
Coverage.
They have a publication precedent system with no real internal structure or management.
Coverage.
We think we're more effective. And of course, we maintain a higher taxonomy as well, so that there are official designations for things like family and order and so on.
Coverage.
First question, yeah, and then another question back up.
Coverage.
For Peter, this is Ryan from NCBI. You commented or highlighted some of the issues with sort of low or species designations that have a high within species diversity.
Yeah. I'm
wondering if, from your perspective, there's any issues in the opposite direction. What comes to mind is begomoviruses, which I like to pick on because of how high the identity is between species. But I'm curious if you think that's a problem or not.
Yeah. I mean, I think there's the aspect of stability. I think, on the one hand, we could say, okay, let's try and make it all 95% ANI, and to hell whether they group together or not. Or else we could say, well, let's take exactly what we have, which is historical, actually, in many cases, and pre-genomic. So a lot of these categories are made before it was known how divergent they were from each other genetically. In terms of stability, there would be, as I showed the alternative classification of enteroviruses, there would be an absolute outrage if you lowered everything by a rank, okay, because suddenly there wouldn't be corniviridae anymore. And I feel that would be highly destabilizing and difficult, and which is why I think actually, if we want to have categories below species, we actually make a below species category so that we can capture some of the clinical entities that we're missing with this very high level that we have, okay? With this very, I know it doesn't make sense from GenBank point of view. It's difficult. With this very, yeah. with this very, thanks.
No comments. We've got a question in the back.
Yeah, Richard Sharman from National Library of Medicine. Derek, so you showed these amino acid substitutions between your antigenic clusters, and yet when you look to see what impact they would have, there seemed to be a very small or single amino acid substitutions seem to have an effect. Is that because of the assay that's used? So if you're looking at effects on hemagglutination inhibition, these other sites might be really important for other immune mechanisms, and that's why you see that they're kind of conserved between these groups. So I want to make sure we're not missing something important in terms of immune mechanisms.
With this
yes. Yeah, this is mostly for virus neutralization, what I showed. And of course, hemagglutination inhibition in flu is a pretty good surrogate for virus neutralization. And there will be other effector mechanisms. It's probably usually the case that VN is a good representation of the correlative protection when there is usually decent-ish, at least, protection. So there's been studies in flu, there's been studies in SARS-2 showing that, the higher titer or titer over a particular threshold is typically protective. And then when the titer as measured typically by VN lowers, then protection will lower. But then it's clear that sometimes when there is extremely low VN titers, there can occasionally be protection from some other effector mechanism, although the protection is typically less. So yeah, there's definitely other mechanisms. And
with this
at least for the viruses that we look at,
with
less
this very
important for protection, but still, when we think about things more broadly, we have to take that into account. And thank you for pointing that out, Richard.
We ran into something like this, trying to develop a universal coronavirus vaccine, where we did ancestral reconstruction and we made, antigens based on those sequences, immunized mice. We got no virus neutralization, and yet the mice were protected from infection.
With this
a very surprising, finding, but I think it just points to other immune mechanisms can play a role.
Yeah, and sometimes that mice can be very broadly protected, And I say that in addition to what you say, not contrary to what you say. Also in flu, mice can sometimes be protected by, transfers that have not worked in other species as well. Yeah.
With this very. Well, let's see another question. We don't have any online questions yet before we continue.
There's one comment. I think it should be why not why extend. Why not extend the ICTV authority downward exactly one level to the subspecies? Why not one or two? Doesn't make adding more ranks below the species, especially for the Brinites problem worse. I'm not sure what that means.
Arbitraryless.
Yeah.
Well, I'm not sure whether they meant why wouldn't you? And I think there is potential argument to do so, given that it's arbitrary, and there are very useful and important categories below species that might be better defined if we had a sort of a taxonomic level to describe them. I mean, obviously, it doesn't solve the HIV problem, but it certainly addresses things like polio, and perhaps SARS-CoV-2 as well. And Elliot, yes, please.
Well, I would just say, though, you do have to stop somewhere.
Yes.
And so we aren't going to try to create subspecies levels for every lineage that we know of for SARS-CoV-2, for example.
Yeah.
So you're always going to have to make a cutoff.
Yeah.
But perhaps you can make that a little bit more intelligently, a little bit more granular than we currently do.
Yeah. And I think there was a proposal going through along these lines then whether you wanted to have a level below, whether it'd be useful or not. I mean, it's the same for biology as well. I mean, lots of species don't have classification below them, but some do because of various practical reasons. And I think it'll be exactly the same for virology as well, where I think the majority probably wouldn't need to be classified below, but obviously you could have that option if you wanted to.
You still want to have a question?
I did. This one's for Derek. A beautiful talk, Derek. I was wondering how good the sera sampling is. And since we have such different, with SARS-CoV-2, for example, such different epidemic histories, so people have been exposed to different variants in different parts of the world, and vaccine histories in different parts of the world, do you think they're consistent and global?
No, I don't think they're consistent in global. And as I know that you know as well, and they will just become more heterogeneous over time, just like in flu. Our experiences is different over time based on when people were vaccinated or infected. And so the sorts of antigenic maps that I drew today were all drawn with primary infection sera. So this is, how the antigenic differences would be seen by somebody who's been infected or vaccinated with just one variant. Of course, when one looks at flu or SARS-2, we're primarily trying to protect at-risk populations that are typically elderly, not always, and they've had multiple exposures already. The way that we think about that, although one can make antigenic maps with such multi-exposure sera, we don't. We make antigenic maps from these primary exposure sera to get these fundamental or basic antigenic differences between strains as they would be seen by people who were being first infected. And then we separately, in a third dimension, if it's a two-dimensional map, and in other ways, if it's a higher than two-dimensional map, we draw those multi-exposure sera on what we call an antibody landscape, which is in an extra dimension. And so you could have somebody, for example, we see that SARS-2 antigenic map, or even that H3 antigenic map from 1968, if we took it all the way through to the present day, I would have immunity against all of those variants. That doesn't make all of those variants the same as each other. It just means that I have protection for different reasons against all of those, which is why we think the right way to make the antigenic maps for this sort of classification is from primary infection sera, and then represent multi-exposure sera independently as these antibody landscapes on top of the maps. The other advantage of that is that then one gets to see tighter magnitude as well, which of course is also relevant for protection. I have a question for Peter, if I may. I'm sorry that in case somebody else had a question there and I jumped in. What was it? Sorry for my language. Yeah. It was mentioned yesterday about having that sometimes there's a separate dimension of how we might want to classify. And of course, the antigenic one is one of those as well. Has there been anything in virus classification or in classification in other areas of biology where people have tried to characterize based on more than one characteristic or to have some extra information in the taxonomy?
Well, yes, of course. I mean, in fact, the original definitions of species were with this and it had multiple criteria, as in the current definition. So obviously we could define a species not just by its genetics, but also we have to know what its sequence is, because we have to assure ourselves that it's monophyletic. But beyond that, then we may want to define it by disease spectra associated with infection, and also antigenicity as well. So obviously there are some species which actually have specific serological properties, like the dengue 1, 2, 3, 4. I mean, that's sort of.
And do you typically encode that in the name, or are you just using it for the?
We have very descriptive names, especially in plant virology. Once you use a name a few times, then the link between what it sounds like and what it means is gone, as we all get used to silly names eventually. That's nominalism. So, obviously, it's they're arbitrary in some sense, but of course it helps if they're somewhat descriptive, just so you immediately know roughly what it is, okay, irrespective of what the definition is. But it's quite variable. And of course, where we now have metagenomic data from, if you get a bucket of water and discover 5,000 new phages in there, the nomenclature for those gets quite difficult, because there's nothing particularly characteristic about those with this sort of pin a name very easily. And of course, there's been temptations to make codes, numeric codes, with this are very unintuitive ultimately. And in fact, names are better, but it's just difficult. But as I saw one way here, I mean, we always have to be encouraged by the entomologists who have classified over 300,000 species of beetle, and they've given proper Latinized binomial names for all of them. And they haven't run out, and they're still classifying now. So, we're only 13,000 species. So, it's from that point of view, it's quite straightforward, really.
A quick comment. This is Anita at NCBI. I wonder if that's not dissimilar to the way that a lot of isolates are named, where you have like the H type, the N type, the country of origin, the host of origin. You could also have something like the antigenic group and the genetic group somewhere in a formatted name.
Yeah. Derek, you really want to.
Oh, yeah. I think that it absolutely is. And, there's times when it's really useful to have those easy to remember names. Yeah.
But I think, one illustration of this is obviously the discussion yesterday, where we have systematic names based on lineage, which get long and collapse and then re-expand and so on. And then somebody at WHO says, well, actually, that's beta, okay, or delta. And with this range, particularly with you, apparently, but resolved when actually we all got used to calling them delta. So, in that sense, the name is somewhat arbitrary. And I say it can be descriptive initially, but obviously it doesn't necessarily have to be fixed that way. And of course, we have lots of virus names which are sort of humans, so blah, blah, blah. But actually, in fact, you find out it infects other animals as well, but you still keep the human prefix to it because that's what its host range was thought to be originally. And you don't have to rename it, but it's just obviously an initial description.
Hi, Tavis Anderson, National Animal Disease Center. Derek, this is a question for your H5 map. If you were to add another dimension of temporal ordering or geography or migratory flyway, would you get better discrimination of the antigenic space?
So Mathilde Richard, that leads the work on that, has done some stuff where she's colored like that. I think it was you who showed some of that stuff in your stuff, right? And yeah, sometimes, as I think you showed, yeah, things are localized in particular regions. Like those clade 1 viruses, the yellow ones being mostly Vietnam from around about 2004. The 2.1s being mostly Indonesia from 2000. When was it? Just in 2005 onwards, something like that. Yeah, and then some of them all more mixed up.
I think we're getting to the end. Is there any online final questions that we should end with?
So there are two questions. One is sort of comment, not a question. Charles is following up with his concern from yesterday. Animal viruses benefits from almost a similar addresses. It gives the example of eptovirus, which includes foot and mouth disease virus, which has 7 serotypes and similar. So that's the comment. And another user Rumini, where the question is, what are the overall potential main reasons for vaccine not to work well for certain individuals? Not related, but just
Yeah, big question. Given the shortness of time, maybe I shouldn't go into it in detail. Yeah, well, it's obviously multifactorial, as the questioner said, with what are the reasons, and I would say also some of the things that we don't know. One looks at flu vaccine effectiveness, for example, in a good year, 50 to 60% of people get protected. That's a long way to go to get up to where we are with measles. And also with SARS-2 today, the SARS-2 at the beginning, way up in the 90s. And today, much lower. So, certainly for flu and SARS-2, one of the things is vaccinating in the face of cross-reactive prior immunity and how much of a de novo response that we get. But there's clearly many other factors as well for pathogens that are not changing antigenically. And those almost certainly apply to vaccinating against pathogens that are changing antigenically as well.